It's an honor to introduce our speaker, a friend, Dr. Christopher Shade. Dr. Christopher Shade is a globally recognized expert on mercury and liposomal delivery systems. He has lectured and trained doctors in the US and internationally on the subject of mercury, heavy metals, and human detoxification system. Dr. Christopher Shade obtained his BS and master's degree from Lay University in environmental and aqueous chemistry. Dr. Shade earned his PhD from University of Illinois, where he studied the environmental and analytical chemistry of mercury, as well as advanced aquatic chemistry. During his PhD work, Dr. Shade patented analytical technology for mercury speciation analysis and later found Quicksilver Scientific in order to commercialize his technology. Shortly after starting Quicksilver Scientific, Dr. Shade turned his focus to the human aspects of mercury exposure and the human detoxification system. He has since developed cutting-edge lipid-based delivery system for nutraceuticals such as liposome and microemulsion system to address the growing need of high-quality, affordable detoxification solutions. He has also developed a specific clinical analytical technique for measuring mercury exposure and a system of products to remove metallic and organic toxins by upregulation of innate detoxification biochemistry. Quicksilver Scientific is the only lab in the US that separates organic mercury from inorganic mercury from the sample. It's an important factor because it is nice to know where a source of mercury is. His current focus is at the intersection of neuroinflammatory issues, immune dysregulation, toxicity, and infection, specifically how to peel away the layers of overlapping dysfunction in the sick individual until you get to a point at which the system rights itself. Without further ado, please welcome Dr. Christopher Shade.
I don't need that. I have lots of amplification going on. Suk is my dentist, by the way. And, okay, wake back up. All right. All right, does that go back up? There we go. All right. So, I know a lot about mercury, more than you'll ever want to know about mercury. I got my PhD in mercury, and it was environmental chemistry of mercury. And at the time, the government wouldn't give any grants to people studying mercury in people, but you could learn anything you ever wanted to know about mercury in fish, birds, different wildlife, what it does in the rain, what it does in the soil, and that's what we did. And so all the best chemists, mercury chemists, were in environmental at the time. And we had tools for predicting what was going to happen with mercury here, there, and everywhere. And I brought some of those tools into the clinical world because, long before I did my PhD, I was, I sort of was disillusioned with science and became an organic farmer. I was a biodynamic farmer out in the woods and, lived in a little tent and stuff. And so I had a holistic mind, but the world threw me back into science. And so I was lucky enough to learn really good science tools to frame holistic thought and really bring that into a world where we had a lot of clinical metals toxicology that was very black box oriented. We'll measure your pee, see what comes out. We'll put a chelator in, and if mercury comes out, that's bad. And we'll give you a chelator, make you get rid of it. And these ideas that you could never get rid of toxins without having specific antidotes to them, like chelators. And the reality is that you have very complex mechanisms in your body for dealing with toxins. You have a lot of stress response mechanisms in your body that are supposed to be able to flush these things away. And it's only really in situations where we get very acutely exposed where we need to move to something like a chelator. So in industrial exposures where it's just really beyond the limits of your body to deal with it. Most of what we deal with in people who are chronically ill are chronic weaknesses in the body's system that are allowing for accumulation of metals and other toxins within the body and within the cells. And so I'm actually not going to talk about mercury today because I'm tired of mercury. Like, I'm done. We have this saying at my house, like, I am so done with mercury. It's just, let's talk about something else. So if you came here for a mercury talk and you're upset, just let me know and I'll just start talking about mercury and you'll feel a little better. But really, I want to talk about the things that I've been into recently, which is how do you turn up these natural processes in the body? And what are the different nutraceuticals that we use and what are they doing? And if you get these sick people that can't take anything, you give them little bits of this and that, and they have Herxheimer reactions. We say, oh, it's a Herxheimer reaction. I'm detoxing too fast, or I'm killing my Lyme and I'm getting sick. A lot of these, they're just toxic from the substance you're giving them because the chemoprotective mechanisms that are stress response mechanisms are broken themselves. And so I'm going to present to you detoxification in terms of the chemical aspects of it that happen in the cell and what we call drainage aspects, how you filter out your fluids. Oh, hey, here's a mercury slide. All right, so there's mercury stuff. It's only the first slide. We're already back to mercury. But this starts to lay a framework for us on how we're all different. I mean, you're here, you're probably aware that we're all different in terms of what we express, both the genes that we've inherited, but then epigenetically, which genes we're expressing at any one time. And what dictates what genes get expressed at any one time. And there's a number of things that dictate that. But in the end, we all end up with different rates of detoxification. And this slide was from a poisoning that happened in Iraq in 1973. And on the x-axis here, you have the biological half-life in days, ranging from 40 to 100 days. And then the frequency of the population. So this is a classic histogram that's showing detoxification rates in these people and their variations. So over here on the left, you have a half-life is a time from a peak level of a toxin in the blood to half peak level. And so over here we have 40 days, and those are the fast detoxifiers. In the middle, we're running around 60 days, and this is for methylmercury. That's your average detoxifier, and you run over here to the slow detoxifiers at 100. Now, of course, I'm ignoring this bar over here, and it's a very big bar. So you have a very classic histogram of population there, but then you have this probably genetic subset of very slow detoxifiers out at 120 days. And so most of the time, if you're running a practice and you have people coming to you with some sort of chronic toxicity issue, they're this group, the slow and super slow detoxifiers. And there's a combination. Everybody's into SNPs these days. You know, what are your SNPs? Oh, I've got a SNP for this or a SNP for that. MTHFR. What do people call MTHFRs? I won't make you say it. I won't say it either. But, are you one of them? Well, I certainly am, Oh, my God, I'm going to die. I'm a 677 double mutant. Like, that's the death sentence, right? 35% of Mexico is 677 double mutants, and like 50% of China. I forget the number. There's a lot of people with that. And they're all right. There's a point at which the whole system starts going down, and you start having changes, and the body starts to get a little bit more down into the muck and having a lot of problems. So those aren't really the SNPs that are happening. These are functional expressions. And sometimes we talk about epigenetics, and I'll talk at the end about specific epigenetics actually blocking expression of your detox genes. But then there's the sort of selective expression based on what kind of situation your body's in. Your membranes are making a lot of decisions on what genes you express. So this is the big Bruce Lipton thing, that the nucleus wasn't the most important thing. The membrane was the most important thing. Because if the membrane was healthy and the membrane read a space around it that was clean, it opted to express genes that were maybe a little bit more of a luxury, like cleanup genes. When you're not in a stress situation, you're not in a survival situation, you can start cleaning up your milieu. So functional expression, and the biggest one with detoxification is inflammation. And I'll show a couple of slides about inflammation and detoxification essentially being on a teeter-totter like this. Because detoxification is part of your antioxidant system. And inflammation is part of a dominantly pro-oxidant system. Inflammation is when something comes in to eat you and you respond to kill it with pro-oxidants. You're making bleach, you're making peroxides, you're making peroxyl radicals. Those are all pro-oxidants. So when you're doing that, you're turning down detoxification necessarily, which means then when you're in chronic inflammation, you're in chronic toxin retention. So that's a big takeaway here. One of the slides, and I have a couple slides in the beginning to just sort of frame all of this. And what are the big points? And then we'll dig in a little bit deeper, and then I'll run out of time and I'll scramble through the last couple of slides. So this is a really good slide because I was teaching the principles of this slide for about 3 years before I found it, and it really nicely summed everything up. And this speaks a lot about the idea of what the body burden is and what toxicity really means, because people always come to me because we have this lab, we do this fancy testing, and they say, oh, I want you to test me and tell me if I'm toxic. So what do I say to them? Our toxicologist might appreciate this. I say, well, if you just stand with me for like 5-6 minutes and talk to me, I'll tell you a lot more about how toxic you are than if I measure you. Because we're not in acute situations, we're in chronic situations. So the question is, how much are these toxins affecting you? And it's easy to pick out, especially with dentists. So in this very important experiment, these are cell culture experiments, and they were testing cells. They had mutated all these cells from this fish that's real easy to work on. And they made cell cultures, and they were looking for a cell type that was resistant to cadmium. And so they found a cell that could swim in cadmium. And then they found it could also swim in mercury and arsenic. So cadmium, mercury, and arsenic, those are big three to remember because they're slaves to the glutathione system. So if you want to get rid of any of those, upregulate your glutathione system. So they found these cells that could swim in all this mercury, cadmium, and arsenic. And so then what they do is they take them apart and they look at what the chemistry inside of them is so they can understand what made it so good in that situation. And they found that there was 3 main things happening. And these are the sort of 3 pillars I teach with mercury detoxification. And one was that it had a lot of glutathione biosynthesis. It made a lot of glutathione. The second was that it had high activity of glutathione S transferase. So if we make an analogy that we're in a cell here, and these are cellular proteins, and we have a mercury bound onto the cellular protein, and we want it to come off. Well, the glutathione swims by, and the mercury doesn't just jump off onto the glutathione. It needs the transferase. It needs a catalysis, catalyzing that transfer. So then that's going to pull that mercury off of that protein. Now we have a mercury glutathione conjugate swimming around here. We have to get rid of it. And so we have a bouncer at the door called the MRP. It's a transport protein that goes through the membrane and moves these toxin conjugates out of the cell. So in this experiment, these cells are able to conjugate, ship away, conjugate, ship away, conjugate, ship away, push away, push away, push away from the cells before the metals would hit the targets that created the toxic response. Now, when they knocked out any one of those, the transferase, the glutathione synthesis, or the transporters, not knocked it out totally, but just turned it back down to a normal level, the cells lost that immunity to the metal. Now, in that petri dish, what we would call the burden, the bioburden, the body burden of those metals never changed. When the detox mechanisms were working overtime, the cells did not have a toxic response to the 5 micromolar cadmium that was in there. But if they turned down that ability to push away all the time, the mercury got to the sensitive sites and created the toxic response and killed the cells. So it's not just that we need to get the body burden down. We need to get the defense up. Now, at the same time that you push the defense up, you will push the body burden down, because detoxification in the body is a microcosm, macrocosm thing. We need to get it out of the cell, and from the cell it goes to circulation, blood, lymph, and then we need to filter the circulation. So there's two parts, the cellular detoxification, and then what used to be called in the European biomedical world drainage. It was lavage in France. And so you have drainage remedies. My mother-in-law, she's French, and she looks at me like I'm crazy with all this stuff, but she will go out and she'll get these little ampules of artichoke extract because she needs some drainage, because that's in her culture. These sublingual nanoliposomes are not in the French culture yet. Soon. All right. A side point on upregulating the glutathione system is that when you do this, obviously we're turning up your natural mechanisms. But glutathione isn't there just to move mercury out of your body, or mercury, cadmium, and arsenic, or any of the dozens of other toxins that it conjugates to and moves out. It's a big regulator of a lot of things in the body. It's the main antioxidant. The ratio between oxidized and reduced glutathione is setting the redox tone in your cells, which is really creating the set points for proliferation versus differentiation. When you have high oxidized glutathione, you're sending out stress responses. Our decisions on which genes to invoke, we're left to a survival mechanism. In fact, there's a lot of really good work now looking at cancer as dropping back to a survival set point. That's easier, it's a way a cell can live that's easier to survive in when it doesn't have a lot of good environment around it. But glutathione strongly controls the levels of TH1, TH2 responses to your immune system. That's innate and acquired immunity. This was a study where they took these mice and fed them a liquid ethanol diet. It's a good thing I have a glutathione company. And the liquid ethanol diet plummeted their glutathione levels. Now, that's not on this graph. What's on here is interferon versus IL-4. Interferon is your main antiviral. It's a killer. It kills the invaders in the intracellular space. The interferon drops to nil, and IL-4, which is more of a hypersensitivity or an allergic reaction, goes way up. So when the glutathione goes down, you start hosting viruses. You've got herpes 1 through 30 just sort of wandering through your system, and you don't touch them, but you're rejecting all of your food. You're becoming intolerant of things that are coming in. So bringing up your glutathione system is not just detoxification. It's normalizing a lot of functions, including the glutathione, including the immune system. Now, I wish this was a little bit more clear to you. I'm going to set up a slide that we'll use a couple times through here as we talk about these aspects of detoxification. And so this is the movement, this is set up to track the movement of a toxin out of the cell through circulation into the liver, GI tract, or into urinary excretion. So you have phases of detoxification, and these are happening inside the cell. You've got transport moving out. You've got phases happening here, here, and here. And I believe, if I'm not wrong, I'm going to go through each of those. It'll be a little bit easier to understand. I just said that detoxification, we can think of it in two parts. Cellular detoxification, remember that conjugation of the toxin onto glutathione and shipping it out. There's other things you can use to conjugate. There's sulfate groups, there's glucuronic acid, there's methyl groups, there's glycine. But the main ones are glutathione, glucuronic acid, and sulfate. So that's cellular detoxification. And then you go into circulation, and you need to filter it out of circulation. And that's going to be the kidney, the liver, the GI tract, and also, Bern, the skin. You've been talking about saunas. So there we go. Kidney, liver, GI, skin. The most classical drainage remedies are kidney, liver, lymph, for lymph movement. And then, GI is definitely one of them. It's just treated as a laxative thing. So moving the bowel. So we move toxins into circulation, then we have to remove them from circulation, with kidney, liver, GI, or skin. The interface in between cellular and drainage is the cell membrane. And the health of the cell membrane is a crucial, crucial thing. I had already brought up Bruce Lipton and his discussion of the membrane reading the extracellular environment, which is the extracellular matrix. I have t-shirts at home say, I love the extracellular matrix. You got to be into German biological medicine to have a t-shirt like that. So in German biological medicine, they treat the matrix, and this is a lot of European medicine, they treat the matrix more than they treat the cell. So let's look at it from that perspective now. The matrix, we used to think of as just this sort of amorphous gel that was outside of the cells. But it's actually this hyper-ordered array. Now you've got these big ropes of collagen going through there, and then you've got these antenna-like arrays that are different proteoglycan complexes, and at the end of those arrays are these things called weak acids or carboxyl groups. And they have to be deprotonated, which means the pH has to be high. When people talk about alkalizing, alkalizing is a matrix phenomena. Acidification of the matrix causes these fibers to lose their charge and they start collapsing upon themselves. So it's like frying an egg and it gets all solid. We think about blood feeding cells, like somehow the blood feeds right into the cell. But the blood has to bring things into capillaries, and nutrients then have to travel out of the capillaries through the matrix to the cell. And all waste has to leave from the cell to the matrix to then diffuse into the blood. And the proper ordering of this is essential. The best work I've seen on what acupuncture does away from the energetics of Chinese medicine and what are the physiological responses from acupuncture is done by the guys at the universities in Germany that do all the German biological medicine. Alfred Pischinger's book, The Ground Substance Matrix and Regulation, I think the name is something like that. They describe all of the events that happen in the matrix after a needle stick. And interestingly, for those of you who are connoisseurs of acupuncture and like Japanese acupuncture where they use very fine needles, they determined that the most radical responses, and the responses moved through the whole body. Those responses, there's only one organ that covers everything, and it's the extracellular matrix. It is connected head to toe. The whole thing is connected. And they see these cascading changes go through there. You got Bill Tiller, who was working on looking at hyperordering of different ion channels through the extracellular matrix. That's where all the energy medicine works. I already talked about the acupuncture. And back to the Japanese thing, they found the most dynamic reaction came from the smallest needles. The Japanese use really small needles. So the matrix is crucial. And the idea of drainage is mostly a cleaning of the matrix. The idea of our fasts and our cleanses is mostly a cleaning of the matrix. And the membrane is that part that mediates between the two. And so we talk about mediation being the membrane deciding how healthy is the ocean it swims in, and then what genes will I express, but also all those transport proteins. So for the transport proteins to work, to actively transport nutrients in and actively transport toxins out, they need fluidity of the membrane. There can't be damage to the membrane. So you need an adequate supply of essential phospholipids, and that is going to allow the fluidity of the membrane because all those transport proteins actually make little changes to their conformation that allow the movement in and out. They need that fluidity. So you can't have oxidatively damaged membranes. You need to be rebuilding those all the time. Incidentally, the best way to get the highest source of phosphatidylcholine is raw egg yolks. That's a great, easy way to go. I mean, you can buy my micellized phosphatidylcholine, but raw egg yolks. Then cutting to the chase of how we turn on the detoxification genes, there is something called the NRF2, which is that little, the Nerf ball, the NRF2. This is in the cell. The circle there is the nucleus. And outside of the nucleus, you've got the NRF2 held in place by the KEAP1. Certain enzyme inducers change the conformation of that protein, and the Nerf ball goes into the nucleus. When it does that, it stimulates transcription of all of the genes that have a certain promoter region called the antioxidant response element. Obviously, it's in response to oxidative stress. Now, a key here, and it's hard to read this, and you have to kind of be into this thing, but you have these two things called SHs there. Those are sulfhydryls. Those are reduced sulfur groups. And in the change in conformation, they're oxidized to disulfides. So it is actually oxidative stress that creates this reaction, and that goes in there and turns on the stress response. And then you remove whatever's causing the stress response. But what if those systems aren't working? So here's a paper, a review of the evidence that ochratoxin is an NRF2 inhibitor. So ochratoxin, one of the worst of the mold toxins, is well known for its nephrotoxicity and renal carcinogenicity. It blows out your kidneys. Blows out a lot of things. It epigenetically blocks the NRF2, so your stress response doesn't work. I was discussing with Bernd earlier, all of these really difficult patients, they come into you, they're so sick, you give them your magic potions for detoxifying them, and they get sicker. So what do? Well, quite often we make up a story, and we say, well, see, it's a Herxheimer reaction because you're just detoxing too fast. Wrong. I just poisoned you with the thing that I gave you, because all the things that we use for detoxification, so say lipoic acid. Is lipoic acid an antioxidant? Say something. No! It's pro-oxidant. See, you knew I was going to do that, so I didn't say anything. But how about green tea extract? That's an antioxidant, right? It's pro-oxidant. And the way that those things work, because they're well-known inducers of NRF2, very well-known, yet I have to oxidize this to get that to go in there. They actually create little free radical cascades in your cells, and they're gentle little free radical cascades, and you turn up your response and you clear them away because you have to detoxify that excessive lipoic acid. And in the course of that, you also get rid of some cadmium, some mercury, some other toxins. But if that switch is broken, I just gave you a pro-oxidant. One of our mutual friends called me up the other day and he said, I just almost killed someone. She's still in the hospital. And I said, what'd you give her? She goes, he said, it was a lipoic acid IV. And these are often awesome. You know, who's Burton Berkson? Bert Berkson does these a lot on cancer and has phenomenal success. You know, like all these guys, they never talk about the ones that crashed and burned. But Steve Hines told me this woman crashed and burned. And I said, did she have mold exposure? And he said, oh, Jesus, she came to me from the head mold guy out in the southeast. And she had massive mold toxin. We were trying to move it out of her liver. I said, well, there's no way to do it. You gave her this massive free radical cascade, and all of her capillaries opened up. She was just like oozing blood through her whole body. So, that's a bummer. And it kind of explains why all the mold toxic people are everything toxic. We say, oh yeah, mold and mercury, they go together, because when your glutathione system is is running too low, you don't detoxify either of those. But then when you look deeper into it, and the mold toxins are actually blocking your ability to detoxify all those. So having given you that bad news, you better hope by the end of this talk I have some way to reverse that. I always say the first half of the talk is Old Testament. It's all bad news. The good comes later. And so epigenetics, who cares? This is just a pretty thing of genes. But, you know, you stick a whole bunch of methyl groups on, and you take the histone tails off, and it shuts the gene down. It stops being transcribed. In fact, that whole body of literature of looking at this hypermethylation of different stuff, a lot of it traces back to prostate cancer. So in prostate cancer, they were sure that this was related to glutathionase transferase SNPs. And then they look at all these guys, they read all their genes, and there's no relationship whatsoever to what your GST SNPs are. You know, so some people gave up on it, and the persistent people said, no, there's got to be. And so they went right after the tumors themselves, or the precursors to the tumors are called pin lesions. And they found in those areas, the genes for glutathione transferase were hypermethylated, meaning they were shut off. There was no activity of glutathione transferase. So then if you're like, you've been tracking this stuff for a long time, remember Dietrich Klinghardt used to say there's a toxin at the heart of every tumor, and they've analyzed all these tumors and they had all these toxins in them. And then they said, well, the toxin created the tumor. But if neoplasticity, the development of tumor, shuts off detoxification mechanisms in that area, then they become magnets for toxins, accumulators of toxins. So often we see a clinical thing, but we don't understand really the mechanism of it. All right, so let's move to talking about these phases here. So detoxification, the reactions of detoxification are described in phases. There's phase 1, phase 2, phase 3. Phase 1 is, let me see if the next, okay. Phase 1 can be described in activation, making something reactive. Phase 2 is conjugation, linking it together with something to make it soluble and transportable. And phase 3 is then that transport out of the system. Now, phase 3 is a big control point. You know, in the phase 1, the cytochrome P450 system, there's dozens of cytochromes. Then you go to phase 2, and there's like a handful. But the transport out, there's really only one. There's one door out of the game. And so it's a very big control point for everything. If you block transport, and those are in the membranes, then you block all of the different reactions upstream from it. So I mentioned phase 1 is known as the cytochrome P450 system predominantly, and this is preparing a toxin for conjugation in phase 2. It's essentially creating a free radical out of the toxin. And notoriously, it makes the toxin more toxic than it was. Now, for metals, there's no phase 1 reactions because metals are already ready to go. They're already reactive. But things like PCBs that were made to be non-reactive, we got to get them out, and so we have to turn them into free radicals. There's a great story in breast cancer and hormones about phase 1 not being matched to phase 3, where certain cytochromes turn estrogen into kind of a radical. It's actually called a quinone. It's a highly reactive estrogen quinone which abstracts DNA base pairs and creates mutations. So in people with cancer, they will measure very high amounts of estrogen DNA adducts coming out in the urine, or people with just high risk factors for breast cancer. Then they found that was true for prostate cancer as well, and they're growing from there. So it's very important to have phase 1 coupled to phase 2. So when you make that quinone, immediately it's linked to glutathione or glucuronic acid, or for estrogen, there's a repair cycle that can bring it back called nitroquinone oxidase, something like that. Those are all phase 2 reactions. And so a lot of this work was done at Johns Hopkins, and they thought about, whoa, we got to make a drug to fix this, and they found that nutraceuticals did that. Nutraceuticals that upregulate phase 2 through, say, that NRF2 mechanism will do that. So phase 2 is that conjugation ability. Of course, they find in the SNP world, people with SNPs for glutathione transferase tend to accumulate more mercury. They tend to have allergies to things like thimerosal. You know, thimerosal that was in vaccines, they used to use that as an antimicrobial, and they'd like wipe stuff down with it. And the nurses who got rashes tend to have GST issues. So phase 3 then is to transport out, and there's a bunch of different names they've given these over the years, but the main names that you'll see now are the MRPs, which stands for multi-drug resistance proteins. They were discovered originally, and the Japanese group that looked at them called them glutathione S conjugate transporters, but then the drug companies were trying to figure out why some people are resistant to chemotherapies, and they have overactivity of this transporter pumping out the chemotherapies from their cells. So they called them multi-drug resistance transporters. And erroneously, people used to talk about phases of hepatic detoxification as if the liver was the only place that you did this. These detoxification reactions have to happen everywhere, but you have multiple copies of that chemistry in the liver because the liver does the lion's share of the detoxification. So it is present everywhere, and of course you have more of it in your excretory areas, so the liver, the intestines, then the kidneys, in terms of how much of this chemistry. So it's not like one cell, one set of transporters. You know, you might have multiple ones. So now to look at it more in a engineering-like schematic, I should put a big pretty membrane right here so that we see that up here we're within a cell, and after that we're moving out of a cell. But in these phases, you've got phase 1, that activation, that free radical generation, moving into phase 2, where you conjugate the toxin with glutathione sulfate or glucuronic acid, and then that transport protein that dumps you from the cell to the blood. But of course, after my speech about the extracellular matrix, you know that it goes from the cell to the matrix, then to the blood. From the blood, then we're in circulation. Now we need to filter the circulating fluids, and that is done through the liver, and there's an active transporter that brings the conjugate from the blood into the hepatocyte and then dumps it from the hepatocyte into the gallbladder, into the bile ducts, and through that into the small intestine. You also have a movement from the blood through the kidneys and transported out through the kidneys. It's important to note that in the kidneys, this is not glomerular filtration. This is active transport in the proximal tubules. They're active, so you've got, in a nephron, you've got the filtration area where the first urine is coming out, and then it's moving through the proximal tubules. And you have active transporters in the tubules that move toxins in and also scavenge stuff that you're trying to save. So there's our more holistic picture, and you can't really see there, but there's the extracellular matrix. You can't see on this woman, but those are not blood vessels, those are lymphatic vessels. So remember, moving lymph is very important. So phase 1 and 2 in the cell, phase 3 moving it out into circulation, phase 3 pulling it into the liver. Phase 1 and 2 have been happening in the liver, filtering and cleaning the portal blood supply. And then phase 3 going out through the bile into the GI tract. You have some transporters feeding the GI tract directly from the blood. And then you have that movement of toxins out through the proximal tubules in the kidneys. I had mentioned that detoxification was part of an antioxidant system, and it's broader than that. I call it the antioxidant detoxification protein repair supersystem. And in this supersystem, at the core, are the things that we think about as antioxidants: lipoic acid, glutathione, vitamin C, vitamin E. Now, all of these things, now including glutathione. So glutathione is a true antioxidant, but if you want it to quench hydrogen peroxide or a lipid peroxide, it doesn't do that spontaneously. It only does it through the action of an enzyme, like glutathione peroxidase. Vitamin C only quenches free radicals through ascorbate peroxidase. once it's done that, it's then oxidized, and we don't want to build up the oxidized forms. The oxidized forms have a negative effect on us, and so we need to reduce them and move them back into the reduced pool, and we have enzymes called reductases for that. So there's dehydroascorbate reductase, there's glutathione reductase. We oxidize proteins and we want to reduce them with glutathione. We use glutaredoxin or we use thioredoxin. These are very important things that control, they're almost like on-off switches for proteins. There's a lot of active sites of proteins that can be turned on or off by glutaredoxin. And quite often if you're under stress, strong oxidative stress, you'll actually cover up sensitive proteins with glutathione. You'll link glutathione onto them to protect them from being damaged any further. And then once the coast is clear and everything gets good again, you move the glutathione off and reactivate that protein. So there's a lot of on-off switch that's happening there. Again, when you're in a stress situation, you turn off a lot of your house cleaning proteins in favor of just survival. Now, the mitochondria have a distinct set of all these enzymes. Inside the mitochondria is a slightly different isoform of those enzymes because they have to live in the mitochondria. The mitochondria are where all of the burning of the fuel takes place, and so there can be some collateral damage from little bursts of reactive oxygen species. So they have to make enzymes that can handle the bursts of oxygen, but the trade-off is that they become more susceptible to damage by mercury, cadmium, and arsenic, which explains why chronic metal toxicity leads to fatigue. Mitochondrial dysfunction and fatigue are hallmarks of chronic toxicity. Last part of our super system is the ones that we talked about. Down here, those are the linking enzymes that link the toxin together with our biomolecules, and up there are the transporters to take them out. Where do you find all the literature for this? This was all developed in the cancer journals. The removal of the free radical generating offender was how it's termed. Now, when's the last time you walked into an oncologist and he talked about detox? Nonetheless, this is where it all comes from. That NRF2 thing that came from mutation research. You can go into carcinogenesis. You can go into any one of the cancer research journals. And the nice things about the cancer research journals is they're open access, most of them, because they've gotten so much public money to do things. All right, so now we go back to our slide here. Now we've talked about a lot of these reactions. And the main ones for the metals, remember, you got to have glutathione, you got to have the linking together, and you got to have the shipping out. And if you're doing that all the time, you can handle being in a toxic world. You don't need to be toxin-free if your cells can adequately keep them away from the inside of the cell. Healthy membranes is something I'd like to emphasize. I brought them up multiple times. Feeding the membranes so that the membranes can catalyze this movement inside and out. And phosphatidylcholine is the main building block, the main of the phospholipids that goes into having a good fluidic membrane. Now, I'm big into Weston Price type nutrition. I think you need a lot of saturated fats, but if you have too many saturated fats, you will have an overly rigid membrane. And so getting phosphatidylcholine from soy lecithin or sunflower lecithin will bring a more fluidic membrane. There's a lot of work in different vegetable oils now. The parent essential oils will lead to a more fluidic membrane as well. So a couple of pictures of membranes and things that are going on. These are transporters that move things in or transporters that move things out. All go through a membrane, and they all have little pulsing. They're opening and closing in these movements, and they need a fluidic membrane around them to support them. And you look at like proton motive force making ATP. You need a membrane that can support a big electric potential across it. It cannot be damaged and leaking. It needs to be intact. All of the energy generation in the mitochondria are through healthy membranes. Damaged membranes lead to leaking of the electron transport chain, creating free radical stress instead of energy. The endoplasmic reticulum is nothing but a big folded over membrane. Boom, 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 boom. If you look at hormone transformation charts, how things move from cholesterol down into all of your steroidal hormones, 80% of the reactions are in the endoplasmic reticulum. I remember the first time I did good phosphatidylcholine therapy, I did lots of injectable grade PC, and it was just like a flush of hormonal energy. I mean, and that was really, I was like, why do I feel so damn good? I felt damn good. And so, I was like, God, my testosterone must be higher right now. And then I saw all these transformations take place on membranes. In the brain, all of these synapse reactions are taking place, all the neurotransmitters are taking place in membranes. These are the transporters in the hepatocytes that move. These are all, it's hard for you to read all these things, but these are all different movements in and out of the hepatocyte, and then everything moving down into the bile canaliculus to drain out through the bile. There's the MRP2, the MDR1. So these are the different toxin conjugates going into the bile. So having the membrane and the transporters work is super important for getting rid of the toxins. But what also happens when the membrane doesn't work, you kill the liver cells. So the other thing that they're transporting into the bile flow is bile and bile salts. And when you have cholestasis, meaning you're failing to produce and move bile, that means that your transporters are failing to move things into the bile. And then you have liver injury because the bile salts, when they build up into the hepatocyte, are acutely toxic. These are the transport proteins, a simplification of the transport proteins in the proximal tubules. Here would be the urine, and there's the blood, and you have things moving in and out. There's MRP2 taking conjugated toxins into the urine, and while at the same time you're scavenging things like amino acids, there's one of Bern's favorite, alpha-ketoglutarate, being scavenged and brought back in. And a more realistic picture is all these different transporters moving in the proximal tubule, moving things into the lumen, into the urinary flow, and moving amino acids and electrolytes back into the blood. When that's damaged, you lose a significant amount of control, not just on detoxification, but on how you scavenge nutrients. So now our picture involves membranes and transporters at the cell level, at the liver level, and at the proximal tubule level. All right, how do we block things? Where are we at here? You wanted me to talk for 3 hours or 4? 5. Bern says 5. 25 minutes. Kidding? I only got through the first 10 slides. All right, we'll have to speed up here. But I got the idea to you. 26. All right, 30 minutes. All right, so now we want to talk about how things get blocked. So I already told you about the inflammation thing, turning things up and down, and I'll just show you one piece of data on that. You'll never be able to read that. So they injected endotoxin into rats. So endotoxin are little parts of bacteria that get into circulation. When they get into circulation, they evoke an acute inflammatory response. Now, it's important to see what happens here because there's a lot of ways that we can get endotoxin into circulation. The most common these days is leaky gut. And when you have damage to the barrier in the GI tract, you move endotoxin into circulation. And here's what happens. So this is transcription of the RNA for the detoxification genes, and it goes 60% down in the duodenum, jejunum, ileum, colon. So your GI detox system turns way down. And in the liver, everything here, these are all different pathways in the liver. They all get turned down except for this guy right here. It's called the MRP3. That goes from the hepatocyte back into circulation to carry things to the kidney to be excreted in the kidney. Now, the significance of that is when you have GI upset, if you have food poisoning, your detoxification through the liver into the GI tract turns way down, and you shunt over to the kidneys. Now, that's fine for a certain amount of time, but eventually you're going to cause damage to those transporters in the proximal tubules. So, now let's watch what inflammation, this is a schematic of how that's all working. We're in the cell, we got phase 1, phase 2, the transport out in the normal intestine. But then when we inflame the intestine, there's a strong downturn in the activity of that liver to GI tract movement. And when that happens, then there becomes a negative feedback inhibition on phase 2 activity. If you can't move out the conjugates that you made in phase 2, you're going to turn down that linking activity. Now, unfortunately, nobody tells phase 1 to turn down. Phase 2 and 3 seem to have feedback controls on one another, but phase 1 seems to come from some other evolutionary pathway. And what happens then is you churn on creating free radicals out of your toxins. The buildup of toxins turns up phase 1, but the inflammation can turn down 2 and 3. That sets you into that situation where you've got inflammation, toxin retention, and internal free radical generation. So that's a picture of chronic disease, and I already told you in the story of breast cancer, that is a picture if you have the cytochromes to make a lot of estrogen-free radicals, where you're going to head yourself towards breast cancer. The lipopolysaccharides I talked about, I'd been talking about this for a while. I found this great paper from like 1992, and this was showing, just hear the title, Concurrent Inflammation as a Determinant of Susceptibility to Toxicity from Xenobiotic Agents. Concurrent inflammation as a determinant of susceptibility to toxicity from xenobiotics. Remember, the load doesn't determine the toxicity, the situation does. And I just showed you that lipopolysaccharides are turning down detoxification. As you turn that down, the toxins are hitting the active sites. And they have this schematic through time. What do we have going on here? I can't even read these. A GI disturbance, boom, you loosen up all those tight junctions, you get lipopolysaccharides into circulation. Alcohol, a little bit of a wild weekend, you're disturbing those tight junctions, you're lowering your glutathione, and you have, boom, you have lipopolysaccharides in there. This was a operation, I think, a UTI infection. So you have these pulses of susceptibility during these periods of high endotoxin in circulation. If you concurrently have high exposure, you're going to have a much more serious issue. So in a normally functioning world, we're going through the liver, we're going through the kidney, but then we go to that situation where we block that liver to GI tract movement, and then we shunt the extra load over to the kidneys. Now that's cool for a while, but eventually we're going to burn out the kidney ability to detoxify, especially if there's a lot of endotoxin in the situation. Now, just to show you the data on that in mouse models, they were looking at here augmentation of mercury-induced nephrotoxicity by endotoxin. So here you have serum urea nitrogen as a measure of kidney damage. Here's your control, there's the mercury. Okay, so mercury is known to be a nephrotoxic and cause kidney damage through free radical stress. Here's lipopolysaccharide alone. It does nothing compared to controls. Now you put equal amounts of the lipopolysaccharide and the mercury together, there's the damage you get. They are synergistically toxic. So what happens then to the ability? over here on the right, you have urinary mercury. Mercury alone, you have high mercury in the urine. Mercury and lipopolysaccharides, you have low mercury in the urine. That's good, right? Low mercury in the urine? Good. Good answer. Where'd it go? Well, here's mercury in the whole mouse. Mercury alone, mercury plus lipopolysaccharides. So you then have toxin retention. So the classical toxicological method of looking at the urine to see if you've been exposed to mercury fails completely. So those were transporters in the proximal tubules that were supposed to be moving the mercury into circulation, and they've been strongly damaged. When we don't have it, I'm not talking about testing today, but when we do mercury testing, we test your blood and your urine, and we separate different forms of mercury so we can see the form that goes in the urine, inorganic mercury. We see that specifically in the blood. If the proximal tubules are working, the urine to blood ratio should be a pretty fixed level for everybody. It's about 7 to 1. But in the sicker people, you see that gets strongly skewed, and the urinary output is almost nil, but the blood levels have gotten very high because you block the drain. There's almost always an infection that has to be treated to fix that. Sometimes it's just a GI infection, sometimes it's systemic, because it was the toxin and the endotoxin together that were creating the damage. All right, so how do we initiate our anti-inflammatory response from inside the body? And this is back to the NRF2 upregulation. This is phytogenomics. Phyto being plants regulating what genes are being expressed, and certain phytochemicals upregulate phase 2 reactions as well as intracellular antioxidants. We sometimes call that the anti-inflammatory cascade. The main groups of chemicals that do that are polyphenolic antioxidants like the green tea extract, pomegranate extract, pine bark extract, and sulfur compounds like you'd have in alliums like garlic and onions, or in the whole broccoli family. One of the more famous ones is sulforaphane. But now we're finding, what does it? Pro-oxidants do it too. So how does that all come together? I think I already told you, but let's go over it all again. So there's that mechanism I was talking about. The NRF2 goes in, and all the chemoprotective genes, the big master switch for chemoprotection, all comes up at once. But it does it through an oxidation of that switch there. So we've got our friend green tea, we've got our friend pomegranate, usually there's a picture of the Buddha, the medicine Buddha at that point. There's his hand, his little blue hand. And the medicine Buddha is holding a bowl of the long life elixir. I'm sure it's a liposome of some sort. I'm not really sure, but I'd like to think it is. But what's in his hand, I know. It's that plant that's called Terminalia chebula or haritaki, and it's one of the best polyphenol sources anywhere. It's all through Ayurvedic medicine. It's known as the king of herbs in Tibetan medicine. It's used to regenerate the intestinal epithelia in Chinese medicine. And you can't see those pictures because we don't have the resolution. But all the things that you'll find in green tea and pine bark, all the great polyphenols are in there. Do I have? Yeah. One of the cool things that's been going on with studies done in mice is they start aging the mice, and they'll expose young versus old mice to something and see how they do. And in this one, they were looking at antioxidant enzymes in young versus old mice. And here's manganese superoxide dismutase, glutathione reductase, glutathionase transferase, all going 35 to 50% low in the old rats. The robustness of this super system has gone down. And then when they treat them with an aqueous extract of the haritaki, all the old rats' numbers, the treated old rats, go back to what the young rats were. Now, what about the young rats? Did the young rats go up 30%? Give me a guess. Great guess. They got nowhere to go. Everything was good. It was, life was good as a young rat. Now we're old rats. It sucks. We're wise but crippled. Sulfur compounds, all the same sort of stuff, vascular protective, anti-carcinogenic, but the sulfur chemicals were a little more prone to intolerance than the polyphenols. So I used to use more polyphenols. The one sulfur compound I use the most is R-lipoic acid. Lipoic acid, and you'll go, like, if you're into mercury stuff, you'll read all the Cutler stuff, and he talks about it being a chelator. There are hundreds of articles on lipoic acid being an NRF2 upregulator of the highest order and being a glutathione system stimulant. And it is specifically the R form that gets into the NRF2. The S form does not do it, and it actually gets in the way. That's why R-lipoic acid is more potent. Those are different rotations. It's like D and L amino acids. You want L amino acids. So in the sulfur compounds, another thing you'll hear about sulfur compounds, people will say, most of you have probably heard that garlic is good for mercury detox. Have you heard that? And people will say it's because it's a sulfur compound, right? And they'll say, and it's because it's a chelator. Wrong. It's not a chelator. These are isothiocyanates. Thiols can chelate, but these are not chelators. They're isothiocyanates. And I'll tell you, they're pretty strong free radical generators. Stronger than the polyphenols. Those are pro-oxidants. Now, what happens in a lot of the toxic people is that they tend to have some combination of upregulation of CBS genes, cystabeta-thionine synthase, which takes all these sulfur compounds and starts metabolizing them to sulfate. But they get built up as sulfite, and they get sulfite toxicity. Sulfite oxidase takes sulfite to sulfate, and it requires molybdenum as a cofactor. So a lot of these people were taking garlic and going, oh, I'm hurting so bad. Wow, man, I can barely see. I was one of them. I did it. I'd take straight sulforaphane. I'd be like, wow, that must be the detox. It was sulfite toxicity, and taking molybdenum totally got rid of the whole thing. So when you're doing the sulfur compounds, you got to make sure that you have adequate molybdenum. Lipoic acid, we can really skip through that. I'll do a couple things on that. Very strong NRF2 upregulator. It is a prescription for type 2 diabetes in Germany. It's used for heavy metal toxicity, age-related decline in the antioxidant system. Remember the young rats, old rats? They did exactly the same thing with lipoic acid. But lipoic acid is a twofer. Not only do you get NRF2 upregulation, you get mitochondrial biogenesis stimulation and more efficiency to the mitochondria. And remember, we've got a lot of damage problems in the mitochondria in chronically ill and chronically toxic people. So that's one of the reasons that we love lipoic acid. This was age-related loss of glutathione synthesis, reversible with lipoic acid. And then also increased metabolic rate because of the mitochondrial stimulation. So lipoic acid Bern was asking me at dinner, well, what's your favorite NRF2 upregulator? I was like, well, lipoic acid. Very predictable, very standardized, and you get mitochondrial and metabolic effects as well as NRF2 upregulation. Ozone. Antioxidant or pro-oxidant? Both. Pro-oxidant going in, stimulating NRF2, generating an antioxidant response from your body. Unless, of course, we have mold toxins and we've epigenetically blown off that switch, which is why some people do very badly with ozone. So it's the same sort of thing, and this is from the ozone papers. And what do they call it? Mild oxidative stress, oxidizing the keep one, allowing the NRF2 to go into the cells and create survival from a stressful situation. And that's just more of the same. It might be a little esoteric, but we were also talking about dinner about ozone therapy, and for a while people were doing ozone and glutathione together. And you ask a chemist like me, or you ask Bernie, you say, you can't mix those two together. You're going to destroy the glutathione. And, you know, doctors, they like a little mechanism, but they're really just like what works, what doesn't. And so they're like, but we get great clinical results. So I'm like, all right, let me think about it. So damaged glutathione is a potent upregulator of NRF2. So they're saying, oh, well, we're giving you glutathione and we're doing the ozone and it's just all great. Now we're giving you this twisted, beat up, destroyed glutathione, which is a stress switch and makes you respond by trying to fix it by turning up NRF2. So that's how that stuff works. And so a lot of, it's basically free radicals are doing this. So pro-oxidants are good. Pro-oxidants are the new rage. You know, it's like everybody's like, well, you saw vitamin E gave everybody cancer. Let's do pro-oxidants now. And you got ASEA, and you got ozone therapy, and you got all these different like bleach-like things. And they're acting as hormetic switches. Hormesis is a stress response. A little irritation, and then there's a big bounce back. So now our list of things that does it, we had the phytochemicals, but really it was the radicals created by the phytochemicals. You've got reactive oxygen species, reactive nitrogen species, reactive sulfur species. These are damaged things. Cascading down and damaging this, and your body says, whoop, let's fix the mess. Let's skip that or we'll never get through. Oh, some good news. How do you clean the brain? So everybody says, oh, make a chelator, go into the brain, get the stuff out. These are the capillaries at the blood-brain barrier, and stained in red is glutathione S-transferase, the linking enzyme, and stained in green is the transporter, MRP2, and in yellow is where they're co-located. Look at that. I mean, that's just like all lit up, meaning all the capillary system has an efflux pump. The blood-brain barrier has an efflux pump there to reject stuff trying to come in and to move out stuff that's in there. But then also remember that inflammation turns down all of those expressions. You get into things like autism. You've got chronic neuroinflammatory disorders. You get into people who have, you give them anything and they have a serious neurological response to it. They've got neuroinflammatory problems. All right, NRF2 upregulation rules. Two things you got to know. You got to have enough substance to do it. You don't trigger these things at small levels. You got to get up to higher levels. Small levels kind of right the ship and bring it from underfunctioning to kind of normal, but you can overexpress it. You can get it up to 200-300% of its normal expressions. And this was using St. John's wort, and this is the control. That's a low, well, let's go over here. Control, low dose, moderate, it's a decent dose. Now we're high dose, really high dose. So at a really high dose, we have a 300% upregulation of the function of the transport protein. But can we do that every day? Can we turn up the system to 300% of its normal functioning and just keep that going? No, there's time limitation on that. And so over here, at that high dose, they got the 300-fold upregulation. This is over 30 days. And it marches over 10 days, up, 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 up. And then for the next 20 days of taking the same dose, it goes down, 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 until you've habituated to it and it stops upregulating. So if you want to use these things to detox, you have to pulse. You want to start at low doses and titrate up to higher doses in pulses. About a max of 10 days on. We start people at 5 days on, 2 days off, and we move up to 10 days on, 4 days off. All right, so blockages. Epigenetic blockage from mold toxins, from neoplastic changes, and I'm sure it's happening and we haven't seen it yet, but viruses. We know there's a lot of epigenetic changes from viruses, but we haven't pinned it down to the kind of things that I'm talking about. But the people with chronic Epstein-Barr definitely have these kind of blockages and need the kind of effects, well, the kind of turning around of those blockages that I'm going to talk about. So there's the matrix and the membrane disintegration. We have to rebuild the membranes and have healthy membranes in order to be able to have the right chemical responses. I won't talk so much about this, but this is when I was talking about neuroinflammatory responses. Martin Pall has done a lot of work on nitric oxide, peroxynitrate free radicals. And this is a hyperstimulation of glutamate receptors in the brain. In fact, the most well-documented neurotoxic effect of mercury is an exaggeration of the glutamate receptors, which is why the most common neurological response to mercury is anxiety. Because you're just like highly glutamate freaked out. And then that moves into these free radical cascades. Then you're foggy and anxious. And then you're so foggy you don't get anything done, which makes you more anxious because you think you're going to lose your job. So then you get depressed about it. So that all starts from having hypersensitivity in the glutamate receptors. And Paul's contention is that multiple chemical sensitivity all derives from these glutamate issues. However, looking back at the whole thing, what if there's epigenetic blocks on NRF2 that are causing excessive free radical damage and you're not able to fix it? And one of the downstream effects is this high glutamate sensitivity or overexpression of glutamate. So we're back to the mold toxin thing. Membranes, let's just blow through. All right. In 10 minutes? Good amount of time. Just to frame how we're going to design detoxification, we need to support the enzyme system, or we need to get that NRF2 upregulation in the cells. We need healthy membranes to support all this activity. And we need a clean matrix. So matrix, we talk about drainage of the extracellular matrix, and we go, what are the European remedies for this? They're bitters. And in fact, we're releasing a bitters real soon that has all of your liver drainage herbs like milk thistle, dandelion, and gentian. Has kidney drainage herbs like solidago, corn silk, juniper, and blood cleaning herbs like burdock, but dandelion's also a blood cleaner. And in Ayurvedic medicine, myrrh was the great blood cleaner, and so we have myrrh in there as well. So, but, you can also think about alkalizing the terrain. The simplest way to go is just good water with bicarbonate. My favorite thing is isotonic sea minerals. There's a product called Original Quinton, Quinton, and they also have a brand called Quintessential. It's a French seawater from a very special place in between the zooplankton and the phytoplankton. It's got a very special phytonutrient blend along with the isotonic sea minerals. And that was used, that was used for 100 years to clean up the terrain. So you need to upregulate the enzyme or the chemoprotective system. You need membranes, and you need a clean matrix. The membranes, I wanted to put this picture before, and again, this is a hard one for you to see. But the electron transport chain is moving through the inner membrane of the mitochondria. And you're turning fuel into ATP and a little bit of free radical coming off of that. A little bit of free radical is good. But when the membranes, and you can't see, there's the schematic here has all these damaged oxidized membranes. When the membranes are damaged, the electron transport chain doesn't line up so well anymore, and it leaks. And now instead of lots of ATP and a little bit of free radicals, you get a lot of free radicals and just a little bit of ATP. So that's another significance of fixing the membranes. Phosphatidylcholine therapy, you can get phosphatidylcholine from eggs, as I said, or you can go to really good nutraceutical sources, either pure we have a pure phosphatidylcholine micelle, or liposomal delivery of different nutrients like glutathione or vitamin C. If you have good liposomes, you're using high quality phosphatidylcholine, and you're able to bring in the nutrients and the phospholipids at the same time. So I got the wrong slide in there because you'll never be able to read that. All right. No, that'll come up at the end. We'll look at it at the end. All right, let's skip to, okay, glutathione supplementation. we need to get glutathione in. Now, if everything was working great, your proteins would be made adequately into glutathione. And if you wanted to just bump that up a little bit, you could put a little extra cysteine in there, either as an acetylcysteine or cysteine or as whey protein. But in the sick situation, your enzymes aren't translating that into glutathione very well, and so you need to get some glutathione in there. For instance, with the ochratoxin that's turning off NRF2, you get rid of that by conjugating it to glutathione. But if you can't synthesize a lot of glutathione, you're going to have a hard time. So there's IV glutathione, but IV glutathione, you don't have transporters to bring glutathione from the blood into the cells very efficiently. Nebulized glutathione is interesting, but more of a local action. It's excellent for chronic obstructive pulmonary disease and emphysema, different oxidative stress in the lungs. You've got transdermal, which has got some effect. Acetylglutathione is a new one on the block that has higher uptake into the blood from the GI tract. It has a higher uptake into cells. And then you've got liposomes. Liposomes are the things that I obsess on, and so that's my chosen way to get that in there. And I've showed you about 1 billion pictures of cell membranes, because phospholipids were used as a therapy in Germany. They made lipostabile, which is an injectable grade phospholipid, and it had phenomenal data on reversing fatty liver disease, changing coronary arterial problems, changing hyperlipidemia, changing neurological problems. It was really a phenomenally useful thing. This is all just by normalizing membrane function. So those phospholipids can be put together into these things called liposomes, where you create a little cell that's a lipid bilayer. They're spontaneously formed when the lipids are injected into aqueous solutions. And if your aqueous solution has vitamin C in it or glutathione in it, it gets trapped inside that liposome. Now, crucial to making a good liposome, you need to make a very small one that will absorb rapidly. I've got a couple of slides of that later. And they found that the ability to get into a cell of a liposome is vastly better than a regular nutrient itself. This was done on your local gin. Redizorb is out of Palo Alto, and that's a liposomal glutathione. And this was done in neuronal cell cultures where they diminished the glutathione until the cells were about to die, and they saw how much IV versus liposomal glutathione they had to put into the culture to bring the glutathione levels back up. You needed over 100 times more plain glutathione, which is what you get in an IV, than you did liposomal. Because liposomes are able to fuse with the cell membranes, and you take the phospholipids into the cell membranes, and you get a much higher efficiency of delivery of any compound that's brought in with that. Now I'm going to skip that one there because it's has a lot going on in there. Let me see. Oh yeah, okay. So we talked about epigenetic blockage. How do we reverse epigenetic blockage? And the best data right now is on the nutraceutical diindolylmethane, methane, DIM. People know DIM is an estrogen metabolite thing, preventing harmful estrogens, keeping beneficial estrogens. But it turns out that, I'll just skip to this one quick, epigenetic modifications of NRF2 by diindolylmethane, and this was in prostate tumors. They found that the prostate tumors are epigenetically modified and turned off NRF2, and DIM was able to reverse that. I started using this maybe 6 months ago, a year ago, and I took all my weirdest, hardest cases, the ones you give them the good stuff, they feel better, then they crash, they feel better, then they crash, or they just feel bad right away. And we started giving them DIM first, and we were able to get them onto a significant therapy really, really quickly. Some of these people were mold toxic. Epstein-Barr was one of them. I don't think this woman had mold toxicity, but she definitely had chronic Epstein-Barr that would just come and go and come and go. She used to be a professional athlete, and she was totally wiped out. A week on DIM, and she was back working out. I mean, it just changed everything. And this is done in a nanoparticle that has extremely high absorption. So I don't know if you're doing it in capsules, how high of a dose. We were doing 20 milligrams twice a day, 20 to 25 milligrams twice a day in a nanoparticle, and we were able to do that pretty quick. So, all right, in the therapy approach of removing the blockage, one, we're going to use DIM for the epigenetic repair. We're going to use something for the matrix. I mentioned the isotonic C minerals. They're one of my favorite, but you're also going to want exercise, water, alkalizing minerals, and bicarbonate to fix the matrix. Phosphatidylcholine for rebuilding the membranes, and vitamin C is also for repairing the matrix. The fibroblasts that build the matrix use vitamin C as a cofactor. It's absolutely required. That's why scurvy, you start getting bleeding of the lips because you stop making the matrix in the gums. And you'll also get free radical control and a gentle detoxification. So now if you're using a high quality liposomal vitamin C, you've got both the phosphatidylcholine and the C together. I mentioned CoQ10 here for restoring mitochondrial integrity. Once we get past the initial blocks, we can bring in lipoic acid too. But the CoQ10 can bring the energy up without creating the detoxification stresses. All right. Yeah, we're pretty close to the end here. I'm going to skip the neuroinflammatory thing and go then. All right, so once you've laid that groundwork, membrane, matrix, epigenetic block, now we want NRF2 upregulation. Now we're going to need a lot of B complex vitamins because you're using those in your detoxification. Now you can start using glutathione without having negative effects. Now you can use your NRF2 upregulators like lipoic acid and the polyphenols. And now you can use the stronger drainage formulas. Some of the really sick people, you can't even give them. I used to use a lot of, there's one place called Soluna Labs. They make really good, what do they call those? Spagyric tinctures. It's sort of a mix between an herbal and a homeopathic, but a lot of the really sick people couldn't even handle those. We had to use total homeopathics, but now they can use, because the herbs were being toxic to them. Once we stop that block, now we can use more intense drainage formulas, and we're going to keep up the PC, C, and CoQ10. Just a little pitch for what we just released a new line. I have a couple brochures of it here. Pure Expressions is a direct-to-consumer line that we make. We also have Quicksilver Scientific, which is our doctor's line. This one is aimed more at these epigenetic changes. There's a nanoparticle diindolylmethane. There's a liposomal C. There's a nanoparticle CoQ10 and a liposomal B complex. You can go online. We just released this line at Autism One, which I just flew in this morning from another conference. I got to fly out tomorrow at 6 a.m. to go back there. I only did this because I already crossed paths with Susan once before and had to cancel. So I knew she would try to kill me either physically or psychically if I didn't show up. So I did come. And she heroically wrote the article that apparently I was supposed to write, but this just missed me. So. And so all these things are liposomes that we do. Just what you got to know about liposomes: to work, they got to be really small. They got to be so small you'll absorb them right to the oral cavity. Because once they get into the GI tract, the bile changes them. So you want ones that you can absorb sublingually. And this has been used in the pharmaceutical industry. They make liposomes all the time, and they're making them all in this small 50 to 100 nanometer range. And this is uptake into cells versus sizes, and we're going from 200 to 150 to 100 nanometers. And if you track these white bars, that's getting into the cell. And it's only when you get down 70, that's 60, 50 nanometers, that's when you get really fast uptake. And that's what we size things as, the same way that the pharmaceutical companies do. That was just some sizing stuff. So all the things that you need for that blockage removal are in the Pure Expressions line, and the hardcore detoxification line is the Quicksilver line. And you can go onto our website. You can get more information about all of that. But with that, let's just wrap it up. Oh, just one note. We did put everything together into something called the Detox Cube that you can get through doctors. And that has a protocol for titrating up, pulsing on and off, bringing in all the aspects of NRF2 upregulation, along with toxin binding in the GI tract that we didn't talk about. So let's end it there and take questions.
Great. Sabat. Thank you. Thank you, Dr. Shade. So questions? Right over here.
So would you want to have the mercury or the testing done first before you even look at that cube for treatment?
Yeah.
Or is it something
There's
that's
chemical and there's clinical indications for where you're at. The testing is great, and it's a great roadmap, especially if you see urine to blood ratios are off. That means that you've got damage to the proximal tubules. Those are the hardest people to detox. Usually means that there's an underlying infection that you have to address. So the testing is great roadmap. But you still need a good clinician to make some decisions about where you're at, and you're going to try. The intro doses of the cube are pretty low, pretty easy to deal with, and you're going to test the waters a little bit. If it's hard for you, you might have to back off and do some other work first. Of course, bringing in the DIM ahead of time is going to erase a lot of difficulty there.
For about how long do you need to do?
You know, we're a little new to this, but it seems to act pretty quick. You're going to do it along with the other things probably for about 2 months. But, you know, even at conferences I go to, these people come up to me and they're like, no way, I can't touch any of this stuff. This one guy I just saw, he says, I can't even hold your bottles, they're too strong for me. And so I got him to take the DIM. Within a day and a half, I had him on everything. So it happened pretty quickly. But I'd say, you know, at the very least, give 2 weeks of DIM would be safe, and then start on the others, and you're going to titrate them up and keep the DIM going, you know, at least 2 months.
So another clinician question. So I've been ill for about half my life, and I'm getting fairly old here. So and I've been doing the box work already, you know. And this sounds so complicated in a way. So I'm wondering how important it is that I find someone who really understands your system and can work more, you know, pretty carefully with me. Or
Well,
is it,
yeah,
you
I mean,
know,
the sicker you are, the more you need a detailed guidance. And yeah, I would.
And how would I go about then?
You can contact Quicksilver Scientific. You'll find our website. There's a phone number there. And you can call and ask who we know in your area that you can work with. There's also consult services. And even if you've got a doctor that doesn't know the system real well, they can call in. We have a professional pharmacist that works for us and does our doctor outreach. If he ever runs into problems, he comes to me, but he knows the system up and down now. And so he'll work with your doctor.
Okay, thanks.
Any other questions?
Hi. I was wondering if you are able to reset the mitochondria, if you've noticed that would have any helpful effect towards any kind of dementia.
Well, it definitely should. It depends what the extent of the damage is. There's a lot of stuff that are going to go into dementia. If you detoxify and reset the mitochondria, you have a great shot at reversing a lot of it. But some is very, you know, really long term, and there's almost scarring going on in the brain. It's kind of hard to fix.
I'm sorry, I should have been more specific. I'm thinking particularly of the issues with glucose not being able to be used.
Oh, yeah, that's type 3 diabetes, where you've got insulin resistance in the brain, and that'll definitely work for that. But you know, the real trick while you're doing that is put them on a MCT, high MCT diet, use that ketone bodies as fuel, you'll definitely be able to work with that. Yeah.
Can you use the indole 3, the I3C?
Yeah, orally, if you're using capsules, indole 3 carbinol turns into DIM in the stomach acid. But if you're doing a nanoparticle that's bypassing all that, you have to start with diindolyl methane. Indole 3 carbinol is actually kind of cytotoxic.
So don't eat your broccoli, not too much broccoli.
Eat your broccoli.
Okay, great. Let's thank Dr. Shade one more time. If you have further questions, we're going to take a 10-minute break. So if you have further questions, feel free to come down and ask them here. We'll resume again at 9 o'clock. So we'll be back then. Thank you.